Welcome to Biology of Marine Mammals. I'm Dr. Leslie Williams, and this lecture will discuss gene movement and how it moves in a population. We'll first start by reviewing what causes genetic diversity through the formation of new alleles. From there, we'll take a look at some of the forces that actually affect the allele frequency within a population. From there, we'll actually move and take a look at two that we have not yet covered, those of gene flow and genetic drift. And finally, we'll spend a little time talking about how marine mammals have been influenced by the concept of genetic drift with respect to a genetic bottleneck. In a previous lecture, we covered DNA mutations, and recall that DNA mutations are actually the source of new alleles. In other words, when you get a mutation in a DNA sequence, you get a different product, and this causes a different look or a trait. In these pictures here, we see snail shells and we see horses that all belong to the same species, but clearly they show diversity with the genetic reflection. That, of course, is going to be because there are different alleles in that population. Now, at this point, you may be wondering, hey, I thought natural selection caused new alleles. Now, that's a common mistake. Lots of people think that, but that's not true. Natural selection is only going to act on the alleles in front of them. They're not actually going to create new ones. Only DNA mutations can do that. But assuming that we actually have some new alleles, let's take a look at the forces that might act on them once they get into a species. We've already seen the first one, and that's natural selection. Natural selection is nature acting on the alleles and picking alleles that are best adapted for that environment. And of course, those become more common in subsequent generations. In a previous lecture, we looked at different types of natural selection, such as directional, stabilizing, disruptive, and sexual. But now let's look at some of the other forces that act on allele frequencies in a population. One of those is called gene flow between populations. This is simply immigration and emigration. In other words, individuals migrating between populations. Another one is called genetic drift. Genetic drift is going to be the one we'll spend most of our time on in this lecture because it has severe consequences for small populations. Let's start with gene flow. Gene flow is actually defined as physical movement of alleles between population. This is a fancy way of saying that you get individuals that are reproductive migrating in between the population. And when they do, they bring their alleles with them and basically contribute them to the next or the different population. When you have high levels of this gene flow, so you get individuals that are consistently migrating in between two populations of the same species, then you actually minimize the genetic differences between the two. This actually keeps those two populations together as one species. If you stop that gene flow between the population, this is actually going to be a contributing force to what is known as speciation, and that's covered in the next lecture. So for now, let's actually skip ahead from gene flow and look at the third force, which is genetic drift. Genetic drift is actually a little bit of a difficult concept. It simply means that the allele frequencies drift or change by chance. Similar to the way you might drive your car. You're driving your car in your lane and you drift a little bit this way and a little bit that way. You're pretty much in the same spot, but you're just drifting. There's no rhyme or reason to it. You're not trying to make a lane change. So genetic drift is random. It usually comes about by chance, where only by chance certain individuals mate, and as a result, we change the allele frequency. In this picture here, what we see is the first generation on the far left, and that's going to be a population of flowers, and some are red, and some are pink, and some are white. But only some of those individuals mate for the second generation, which is the middle column. Now, in this picture here, we have a very interesting scenario. We're going to take two red individuals and allow those to be the only two to mate. Just by chance. It just happened to play out that way. It's not that nature selected the red flowers or that the white flowers all died. It just, maybe that's the ones that just happened to find the right pollen flower combination and they mate. Now notice what happens in the final generation. All the offspring are going to be red. This means there's no more white or pink flowers. We have lost the alleles for those flowers. So through random chance, individuals mate. We have drift in the allele frequency, and this can lead to something called gene fixation when the population is small. Now that's a really key idea, when the population is small. When the population is small and there's only a few individuals that are actually mating, gene fixation, which means all individuals share the same allele, there's only one allele in the population at this point, happens really quickly. When you have a really large population, there is genetic drift, but it's not pronounced, and usually we consider it to be a negligible force. Now, if you have a small population and you move in such a way that you actually get gene fixation, which means there's only one allele for the entire population, that's bad news. That's not good for the adaptive health of that population. So here's why. Once you lose an allele, for example, the white allele in this flower population, you can't bring it back. You can't say, hey, I really want the white color now because I think it's advantageous for some reason. It's gone. You have to wait for a whole nother DNA mutation to bring it back. That could take thousands and thousands of years. So clearly, gene fixation reduces the genetic diversity substantially. Okay. Genetic drift, which is basically the cause of this gene fixation, is going to be most pronounced in two situations. One is called genetic bottleneck. In this case, in this picture, you imagine that you have a bottle, and in each bottle is basically a bunch of different colored marbles. Each marble represents a different allele. A bottleneck, you may have heard the term bottleneck before, such as a traffic bottleneck, basically is where you basically turn over the canister and only a few drop out, and it's random. It's just a random few, and the rest basically get put away. They don't get to leave. And that is what we mean when we say a bottleneck. In a population, what this means is only a few individuals from a very, very large number actually reproduce. We see this most often when we see basically a near extinction situation in a species, a rapid population reduction. It can be due to a climate change, such as very frequently it happened during the ice ages, or it can be due to man. And right now, that's what we're seeing. In marine mammals especially, we're seeing a lot of genetic bottlenecks because of man basically hunting whales and seals. And as a result, bringing their numbers to very, very, very small levels in a very rapid amount of time. So that's one example of where genetic drift, once you have that very rapid contraction of your population, is going to have big swings and big consequences. So again, a genetic bottleneck is where you basically take a big population, you cut it down to size to only a few individuals, and it was kind of a random few individuals, and as a result, you lose the rest of the genetic diversity. Another example is called a founder effect, and we do see this too, but not nearly as much with marine mammals. And a founder effect is basically where you take a small subset of an existing population, you move them like to another island, and they start a new colony. And of course, because you're just selecting a few individuals, you lose your genetic diversity on that new island. Okay, I mentioned before that low genetic diversity is a bad thing, and of course, low genetic diversity comes from gene fixation. Gene fixation is basically a consequence of genetic drift in a small population, and one example of that are these genetic bottlenecks. Unfortunately, marine mammals in the last 3 centuries have been extensively hunted by man. These include both whales, which get a lot of press, but also seals. Even though sealing isn't nearly as common now as it once was, we have actually made several pinniped species extinct due to our hunting activities. And many seals and other pinnipeds have been reduced to such low numbers that they have clearly experienced a genetic bottleneck. Probably the first example to come out of new DNA technology to say, hey, where are we seeing these genetic bottlenecks, is actually in the northern elephant seal, which we can see along the California coast. Now, the northern elephant seals actually represent something of a success story because currently population sizes are well in excess of 100,000 individuals. So think about that for a moment, 100,000 individuals. This is actually pretty substantial, considering that earlier in the 20th century, so in the early 1900s, they were reduced to about 20 individuals or possibly less than that. In fact, we thought we had made them extinct, except that a few individuals actually remained on Guadalupe Island off Mexico. So it's estimated that it's about 20 individuals have since been the founding members of the current 150,000 plus elephant seals. So clearly they're a success story. So you might ask yourself, okay, they've reproduced successfully. Why is this an issue? It's an issue because they do not have the genetic variability that is needed should the environment change. With cheetahs, which have also gone through a genetic bottleneck, we've noticed that individuals that are not even related to each other can accept skin grafts, and that means that they're very similar genetically. But also, they don't seem to have the genetic diversity that helps them withstand viral infections that end up being extremely morbid or having a high mortality rate. It could be that these northern elephant seals are going to be in that same boat soon. Because they don't have the genetic diversity, they're not able to adapt to a changing environment as readily as more diverse species would be able to. Okay. So probably many marine mammals have actually gone through these genetic bottlenecks. It's a little easier to actually pinpoint in seals and sea lions because they come out on land than it is in cetaceans, but don't be mistaken, they've happened there as well. So because genetic bottlenecks due to our exploitation of them have been so pronounced in marine mammals, let's kind of take another look again at how all of these concepts relate. So we started with the idea that genetic drift will change allele frequency. It's not usually pronounced unless you have a really small population size. Now this has happened with many marine mammals as a result of hunting activities, such as genetic bottlenecks. If you get a very rapid reduction in population size, you're much more prone to this idea of gene fixation, whereas you get one allele for the gene. There's no diversity among individuals in specific genes. When you lose this genetic diversity, your population or your species as a whole basically doesn't have the resources for which natural selection can act on and allow that species to adapt to changes. Now, I mentioned the northern elephant seals are a success story. That seems to be more rare than what we'd like to think for the rest of the marine mammals. Many marine mammals that have gone through a genetic bottleneck are currently having a lot of difficulty recovering their population stock size, and as such remain endangered or critically endangered. This is probably true for certain populations of the gray whale, the right whale, and it's also true for the Hawaiian monk seal. The Hawaiian monk seal has also gone through a severe genetic bottleneck. In fact, it's probably more severe than even the northern elephant seals are, and they are having a lot of difficulty recovering. In fact, their numbers are currently around 1,000 to 1,200 individuals, and their reproductive success is not as high as we'd like. So could it be that this genetic bottleneck has been a negative influence on their recovery? Maybe so. Okay, thank you very much, and I'll see you next time.